Hello students, welcome back to biology classes. In last video, we had discussed regarding the types of mutation that occurs in plants as well as in animals. In today's topic, we shall be discussing the various genetic disorders that takes place in human because of mutation. The first study in human genetics was carried out by a British Dr. Sir Archibald Garrod in 1902, and he discovered the black urine disease called Phenylketonuria. This was the first study of inborn error of metabolism and biochemical genetics. Therefore, Sir Garrod is popularly known as father of biochemical genetics and also the father of human genetics. He observed that Phenylketonuria is caused by a recessive gene that is inherited like Mendelian factor, that is from one generation to other. Phenylketonuria is a rare genetic disorder characterized by the accumulation of an acid known as homogentic acid in the body, leading to dark colored urine that turns black when it is exposed to air. Sir Garrod and William Bateson in 1902 reported several disorders that was seen in human population that seemed to be inherited like Mendelian gene. So from here, the study on human genetic disorders started. Disorders can be grouped into two categories, namely Mendelian disorders and chromosomal disorders. Mendelian disorders are transmitted from one generation to the next generation following Mendelian principles of heredity. So therefore these disorders are called as Mendelian disorders. These are mainly single gene mutation, and this mutation can be dominant or recessive one. When we speak about single gene mutation, it can be a segment of DNA that has undergone mutation, or in a gene or in the sequence of DNA, any nucleotide or the base pairs are mutated. This mutation can be autosomal or sex-linked. That means the mutation can occur on autosomal chromosome or it can be on the sex chromosome. Accordingly, various disorders are seen in the human population, namely Phenylketonuria, Thalassemia and sickle cell anemia coming under autosomal linked genetic disorders, and hemophilia and color blindness come under sex linked genetic disorders. So these inheritance of Mendelian disorders can be represented in the pedigree chart, as they are transferred from one generation to another. And by knowing the nature of the defect due to these disorders, appropriate steps can be taken to minimize, and also to counsel the couple in order to eliminate these disorders. So today in this video, we will discuss various Mendelian autosomal linked genetic disorders, namely sickle cell anemia, Thalassemia and Phenylketonuria. The first one is Phenylketonuria, which is autosomal linked recessive trait. That means this trait occurs as a recessive gene on the autosomal chromosome. Phenylketonuria, namely PKU, is observed in an individual when the parents are heterozygous or carrier of this disease. The heterozygous parents are normal, but they are carrying this disease, and therefore it leads to the phenylketonuria in one of the offspring. Since it is recessive disorder, that means parent should have recessive allele in them, right? In the heterozygous condition. Therefore, the one of the offspring will express this trait, okay, when both the recessive alleles are present. So, phenylketonuria is an autosomal linked recessive trait. It is an inborn error of metabolism. That means since birth, this disease or disorder is seen. It is also called as single gene mutation. That means only a gene defect will lead to phenylketonuria. That is, alteration of one gene will lead to this disorder. That is located on chromosome number 12. The gene that is responsible for the release of an enzyme called phenylalanine hydroxylase enzyme. So, the affected individual will lack this enzyme since the gene is mutated. So, what is the role of this enzyme in human body? Phenylalanine hydroxylase converts or catalyzes the conversion of an amino acid, namely phenylalanine, into another amino acid, that is tyrosine. Consequently, this phenylalanine is metabolized into phenylpyruvate and other derivatives. Tyrosine helps in brain development and also improves the alertness and attention as well as to focus, and all the brain chemicals such as neurotransmitters as norepinephrine and also epinephrine as well as dopamine, which are very much essential. So these chemicals help the nerve cells to communicate and even regulate the mood. We already know the role of these neurotransmitters such as norepinephrine and epinephrine, that they are also called as natural fight and flight responses to the stress, that are released by adrenal medulla and the nervous system. And dopamine is called as happy hormone, which is essential for mental, emotional as well as to carry out various motor reaction. So lack of the enzyme phenylalanine hydroxylase will lead to accumulation of phenylalanine, and therefore it will lead to the disease or disorder called as PKU or phenylketonuria. Accumulation of these amino acid phenylalanine in the brain will lead to the damage in the brain as well as nervous system, leading to mental retardation. Therefore, there will be also intellectual disabilities and the stunted growth. These individuals have musty odor in the breath as well as in the skin as well as in urine too, that is caused by too much of phenylalanine in the body, and also they will excrete more amount of urine. They have reduced hair and fair skin as well as blue eyes because the phenylalanine can't be transformed into melanin pigment that is responsible for the hair color as well as skin color. They have uncontrolled body movements and also they have behavioral, emotional as well as social problems. Next disease is Thalassemia, that is autosomal linked recessive blood disease. Indicates that this disease is due to the recessive gene located on the autosomal chromosome, and the disease is with respect to the blood. So this disease is transmitted to the offspring when the both parents are carrier of the disease, or the parents are heterozygous. So it is an inherited disease of faulty synthesis of hemoglobin leading to abnormality in the blood. So therefore it is called as a blood disease. Our RBCs are composed of hemoglobin, and hemoglobin is comprised of heme and globin. Heme is very important prosthetic group that mediate reversible binding of oxygen by hemoglobin. And globins are the proteins that surrounds and protect the heme molecules. Globins are of two kinds, alpha globins and beta globins. This is a structure of a hemoglobin wherein there are two alpha chains and two beta chains. That means two alpha globin chains, these are the two, and there are two beta globin chains that protects the heme units with iron atoms. There are the genes on the chromosome that is responsible for the formation of these alpha as well as beta globin chains of hemoglobin. But if there is any mutation or deletion of this gene, will result in the reduced rate of synthesis of one of the globin chain of hemoglobin, that will lead to abnormal hemoglobin leading to anemia. Therefore, result in the disorder called as thalassemia. So therefore, the individual who is suffering from thalassemia will produce malformed RBCs in their body. So depending on the globin chain that is affected, there are two types of thalassemia. Namely, alpha thalassemia indicate that alpha globin chain is defective, and beta thalassemia indicates that beta globin chain is defective. Alpha thalassemia is also said to be A- chain or A- thalassemia, and beta thalassemia is also known to be A-cap thalassemia. Alpha thalassemia is controlled by two genes, HbA1 and HbA2 globin chains, and these two genes are linked together on the chromosome number 16. So the genes are closely linked together. Okay? Whereas beta thalassemia is controlled by only one gene, HbB, on chromosome number 11. So these both the chromosomes are located or called as autosomal chromosomes. So alpha thalassemia is caused due to the mutation or deletion of these genes. Okay? So altogether there are 4 alleles that are present here, since there are the 2 genes, right? So if there is the deletion or mutation of one or more of these 4 alleles, okay, it will lead to reduced rate of alpha globin chain in the hemoglobin. Therefore, there will be less number of globin synthesis. Okay? And beta thalassemia will be caused due to mutation of one or both these alleles. Okay? And therefore, beta globin chain production will be reduced. So, it depends on how many number of genes are deleted, okay, or how many number of alleles that are deleted from that chromosome. More the number, less globin chains will be produced. So due to alpha thalassemia, there will be deficiency of A dash globin chains or alpha globin chains. Therefore, we can say in this case, beta globin chains will be more in number, but it will bind the oxygen poorly, and therefore what will happen, there will be low concentration of oxygen in the tissues, as A dash or alpha chain, globin chain, play important role in binding the oxygen. And the individual suffering from beta thalassemia will be having less number of beta globin chain or A cap globin chain. So therefore here alpha globin chains will be more in number, but they will form insoluble aggregation inside the RBCs, and that will lead to coagulation of blood or clotting of blood, that will lead to death of RBCs, causing severe anemia. And therefore this disorder is also called as quantitative problem with respect to the number of globin chain. That the symptoms that we see in thalassemic patient are fatigue, weakness or shortness of breath, that means they have difficulty in breathing. Then due to jaundice, their skin is yellow in color as well as the eyes. Then they also have deformities in the facial bones, slow growth and swollen abdomen, and also they release dark urine. The next disorder is sickle cell anemia, which is also an autosomal linked recessive blood disease. Okay, so it is the disease which is caused due to recessive gene located on the autosomal chromosome. So the parents who all are carrier of the disease or heterozygous parents, they transmit this disease to their offspring when both the parents have one gene having this trait. So sickle cell anemia is controlled by the HB, also called as HbA and HbS. The normal one is HB or HbA. Okay. So the individual who is suffering from sickle cell anemia will have HbS and HbS in them. So when the cross is made between two heterozygous parents, those are the carrier of the disease and they will be normal individual. Okay. So when the cross is made, we see that 1/4 of the offsprings are normal. Half of the offsprings will again be the carrier of the disease. As I said, we can call this normal gene as HbA2. Okay. So HbA or HB we can say. So the carrier of the disease, there are two offsprings or half of the offspring having HbA, HbS, right? And 1/4 of the offspring will be the diseased one having HbS, HbS. So there are 50% chances of transmission of the mutant gene in the progeny. We can see here 50% chances are there. Okay. So our RBCs have hemoglobin, okay, that is the red protein that transport oxygen from the lungs to all parts of our body, right? And this is because of a gene that tells our body to make iron-rich compound that will make our blood red and which will enable for the transportation of the oxygen to various parts of our body. But if due to some reason, if there is mutation in the gene leads to abnormal hemoglobin causing RBCs to change their shape as sickle from the concave shape. And this happens due to polymerization under low oxygen tension that changes the shape of the RBCs from biconcave cells to sickle-shaped elongated cells. These sickle cells, hemoglobin, they release oxygen due to tension and therefore they tend to stick together. We can see in this picture. So these hemoglobin molecules becomes abnormal and they stick together and forms long chains of polymer strands, due to which the cells get elongated and becomes rigid, different from that of the normal RBCs with the hemoglobin molecules. This happens due to a mutant recessive allele that is located on the chromosome number 11, which is the autosomal chromosome. The DNA that is responsible for the release of beta globin chain or beta globin proteins get mutated in chromosome number 11. Change in one nitrogen base leads to change in the mRNA. We can see here change in one nitrogen base will change the sequence of mRNA and therefore it will change the sequence of beta globin chain. Okay? When mRNA get translated into protein chain, that is the chain of amino acid or peptide chain. So this is the normal beta globin which was supposed to be formed by the DNA. So if there is any change in the sequence of DNA will change the sequence of mRNA and therefore abnormal beta globin chain will be formed. So the amino acid which was supposed to be formed in normal beta globin, okay, is not formed here. We can see in the mutant beta globin chain, right? And this happens because of one nitrogen base. We can see here, yes. So here in mRNA we can see adenine that is replaced by U, right? And this is because of the mutation that has occurred at this site on DNA sequence. This mutation is also called as point mutation since there is the substitution of only one nitrogen base or mutation of only a single nitrogen base. Okay. So here the mutant gene causes the substitution of glutamic acid, okay, by valine. This is the another amino acid. Okay. In normal beta globin peptide chain, there was glutamic acid which was supposed to be formed is now substituted due to mutation into valine. Okay. And this happens at the 6th position of the amino acid chain or beta globin chain where the mutation has taken place. And this happens because of the result of single base substitution from GAG to GUG here, right? Only because of one change in the nitrogen base causes the mutation in the beta globin chain leading to sickle cell anemia. The normal HbA peptide chain, beta globin chain of hemoglobin, okay, leads to abnormal HbS beta peptide chain of hemoglobin. The abnormal hemoglobin, due to the strands that are formed inside them and due to its shape, it blocks and forms the clot in the blood vessels. Therefore, it obstructs the way of the blood in the blood vessels. So, there is no free flow of these RBCs into the vessels here. So, the various symptoms that we see in the patients suffering from sickle cell anemia are the change in the RBC shapes from biconcave disc to elongated sickle-like structure, clotting of these RBCs and also degeneration of sickle cells will lead to anemia since they can't pass easily through narrow capillaries. These sickle cells, they die in 10 to 20 days. Then the normal RBC cells that usually live for 120 days. So therefore, shortage of RBCs leads to this disease. The patient becomes anemic. The body gets deprived of oxygen due to reduced circulation as it blocks the way of the blood. Due to improper blood flow into the hands as well as feet, we see there is the swelling of these hands as well as feet of these patients. Weakness, headache, jaundice, heart failure as well as kidney failure, etc. are the other symptoms that we see in these patients. So we see here that a gene deformity or defect in one nitrogen base mutation or nucleotide mutation leads to multiple phenotypic effect or expression, right? So therefore we call this abnormality or the disorder as qualitative problem, as one gene defect is responsible for all these symptoms. So this was all about Mendelian autosomal linked genetic disorders. Okay, I hope you all must have understood this topic. Thank you.